Next, we're going to be looking at what's happening to the sarcomere during the cross-bridge cycle. So what's happening is we get more overlap between actin and myosin. So that means that the thin filament or the actin myofilament is going to slide centrally, meaning that the Z disc are going to be pulled to the M line. We're going to see shortening of the I band, the distance between the Z discs shorten because the Z discs are being pulled toward the M line. The H zone, where we had just thick filament within the A band, is going to disappear, meaning the contiguous A bands move closer together. So we can see in this image on the left that we have the relaxed version of a sarcomere, and then we have the contracted version. So we see no more H band, we see more overlap between actin and myosin, or thick and thin filament. So we have a much shorter sarcomere during the contracted phase as opposed to the relaxed phase. But we have no shortening of the filaments themselves, just more overlap. So it's very important that you know that actin and myosin, the myofilaments never shorten, we just get increased overlap at the end of the cross-bridge cycle. So here's just another image showing you the changes in appearance of a sarcomere. We have the sarcomere at rest on the left, the sarcomere slightly contracted on the right, so the filaments are still sliding past each other, creating more overlap. And this is what would happen to the myofibril made up of sarcomeres inside the cell. So here's one myofibril in its relaxed state, and it shortens because its sarcomere shortened during contraction. And if that was happening in the entire cell, the cell would shorten because all its myofibrils shorten. And if all the cells in a muscle shorten, the muscle shortens. And the role of calcium as the final trigger for contraction is very important for this relaxed to contracted state of the sarcomere, because it is calcium that initiates the cross-bridge activity by moving the troponin and tropomyosin cascade out of the way, allowing myosin to bind to actin. So it's calcium that binds to troponin that initiates the cross-bridge activity, or the sliding filament model of contraction. Because cross-bridge activity relies on calcium, it actually ends when calcium is actively pumped back into the sarcoplasmic reticulum. Actively pumped back means that ATP is required for this process. Next, we're going to be looking at how to trace an action potential, because that indicates the changes in sodium-potassium ion channels as they travel in and out of the sarcolemma. So just in general, for a cell, there's going to be a greater abundance of sodium ions outside the cell and a greater abundance of potassium ions inside the cell. And that creates a certain type of voltage difference or membrane difference across the sarcolemma or across any plasma membrane. But that means that the plasma membrane of all cells is slightly polarized. In a local condition, meaning just looking at one cell, we would say that the inside of the cell is more negative than the outside of the cell. And that again is just a local summation, because if you look at all of the charges within the human body, they would actually cancel each other out. There'd be about the same number of negative and positive charges. But if you're looking at just one location, we can compare two sides of a membrane or of a boundary, which is a voltage difference, and basically measure them and figure out, is one side more negative, one side more positive? Is it because of the concentrations of chemical ions across that membrane? And we found out that indeed there is a local voltage difference across plasma membranes, and specifically for things like nervous tissue and muscle tissue, their cell membranes have an important voltage difference, wherein the negative inside, if changed, can create an action potential in that membrane. And an action potential is really just the result of a predictable sequence of electrical changes across a plasma membrane. So if we can disrupt that local charge of a negative inside and positive outside plasma membrane, then we can create an in-plate potential and then possibly create a longer depolarization signal and then reset the cell via repolarization. So the 3 steps involved in creating an action potential begin with that local change at the junctional folds of the sarcolemma. So generation of an in-plate potential is a local voltage change that occurs when acetylcholine binds to its receptors and opens those ion channels and allows for a quick influx of sodium ions into the cell. Depolarization is the prolonged result of generating an in-plate potential. Depolarization travels along the sarcolemma, and this depolarization essentially generates and propagates an action potential. And this is due to the opening and closing of sodium and potassium channels. And the repolarization is restoring the sarcolemma to its initial state before we generated an in-plate potential. So this is a reset stage in order for the muscle cell to be able to be signaled again. So here are the steps of generating and propagating an action potential along the sarcolemma of a plasma membrane. So we're starting at a location that should be familiar, which is the neuromuscular junction. Here is an axon terminal filled with vesicles of acetylcholine. Acetylcholine gets released via exocytosis and binds to acetylcholine receptors. When the acetylcholine receptors open, they allow for an influx, or sodium, to enter the cell, and an efflux, or potassium, to exit the cell. More sodium enters than potassium exits, and this disrupts the charge that is normally negative inside the cell and positive outside the cell, creating a positive inside environment. And then that depolarization, which at first is local, so this is a generation of a local end plate potential, will be able to travel and influence protein channels further along the sarcolemma. At this point, I recommend that you watch an A&P Flix video called Neurophysiology Part 2, Generation of an Action Potential. Here is the link, but you can also find this video by Googling its name or looking for it on YouTube. This is not a required video, but if you would like to see the generation and propagation of an action potential in real time, or slowed down time, but with real explanations, you can look at this. This has to do with a neuron specifically, but is similar in the sarcolemma. So let's go through the steps for generating an action potential in order to initiate muscle contraction. So this is generating an action potential in the sarcolemma of a skeletal muscle cell. We have to begin with the nerve impulse traveling from the axon terminal to the junctional fold of the sarcolemma, which is only possible because voltage-gated calcium channels open up in the axon terminal membrane, and when calcium enters the axon terminal, acetylcholine leaves via exocytosis into the synaptic cleft. So that's what we're seeing here for step 1. Voltage reaches the axon terminal, which would open voltage-gated calcium channels. So it's left out of the image, but calcium would enter the axon terminal, causing acetylcholine to exit via exocytosis, and acetylcholine would enter the synaptic cleft. The released acetylcholine would then bind to its receptors, which are on the junctional folds of the sarcolemma. When they bind to their receptors, the receptors open, so they're gated channels, and they were just waiting for acetylcholine to bind. And once acetylcholine binds to the acetylcholine receptors, the gates open, and they're sodium-potassium gated channels, so when they're open, they allow for sodium and potassium to move through them. We get a greater influx of sodium, or sodium moving into the cell, than potassium leaving, and this great influx of sodium disrupts the normally negative charge inside the cell, because sodium has a positive charge, and this changes the local voltage difference across the cell membrane, creating a local voltage change called an in-plate potential. At this point, this initiation of the action potential is just happening at the neuromuscular location. So the creation of a end-plate potential is a local depolarization caused by the influx of sodium, which is possible because acetylcholine bound to its receptor in the junctional folds of the sarcolemma. All right, the next step after the local depolarization has opened voltage-gated channels and the sodium has moved into the cell, disrupting or causing a local voltage change, is the generation and propagation of an action potential. We want this signal to travel from the local location where the neuromuscular junction is. We want it to travel along the rest of the sarcolemma. As it travels along the rest of the sarcolemma, essentially it's a traveling band of charged ions. It's going to cause other ion channels to open. So the transmission of the action potential along the rest of the sarcolemma and to the T tubules is going to cause sodium and potassium channels along the sarcolemma to open, but eventually are going to cause the ability of this action potential to reach the T tubules. And inside the T tubules is a voltage-sensitive protein, and when that voltage-sensitive protein comes into contact with a voltage, it stimulates the opening of this voltage-sensitive protein, which is a channel, and causes the release of sarcoplasmic reticular calcium into the sarcoplasm. And releasing calcium into the cytosol is the final step for contraction, because that rise in sarcoplasmic calcium is going to trigger the removal of the tropomyosin blockade, because we're going to see that the calcium binds troponin, and when troponin moves, tropomyosin moves, and that allows for the CRISPR cycle to occur because it allows myosin to bind to the actin active site. So let's just review those steps. Voltage reaches axon terminal, causes voltage-gated calcium channels to open. Calcium goes in, acetylcholine goes out, leaves the axon terminal, goes into the synaptic cleft, travels across the synaptic cleft, and binds to receptors in the junctional fold of the sarcolemma. The receptors are channels. When they open, we get sodium entering the cell, potassium leaving. The greater influx of sodium creates a local voltage change called an end plate potential. This end plate potential is prolonged via the propagation of this signal, or of the positive ions that entered, and this positive ion travel is going to propagate or make the movement of the action potential travel along the sarcolemma. As this wave of depolarization of positive charge moves past separated sodium and potassium channels, it opens the sodium channels and allows for even more sodium to enter the cell, which creates a continued wave of depolarization that travels all the way to the T-tubules, down the T-tubules, and allows for the release of calcium from the sarcoplasmic reticulum thanks to another voltage-gated protein. Now what we're seeing, or what we just saw, was the stimulation of one single muscle cell. Now this would have to happen in multiple muscle cells in order to generate enough force to, say, pick up a large barbell. For that reason, each muscle is served by at least one motor unit, but normally there are plenty of motor units going to a muscle. A motor unit is just a somatic motor neuron, so the motor neurons we've been talking about with axon terminals, and all the muscle fibers within a muscle organ that it supplies. So let's look at this motor unit. We have one motor neuron, but it has 3 branches at the end, so its 3 axon terminals touch different skeletal muscle cells. So this motor unit would actually be made up of one motor neuron and 3 skeletal muscle fibers. So the 2 parts of a motor unit are the motor neuron and the muscle fibers that are innervated by that motor neuron. If you use more motor units compared to less motor units, you generate more force as compared to less force or lighter force. So let's just look at the anatomy again for a motor unit. These are nerve muscle functional units. So here we have motor neuron 1. It has axon terminals going to 2 skeletal muscle fibers. Motor unit 2 has axon terminals going to 3 skeletal muscle fibers. In order to contract this muscle, we could utilize both motor units or just one, or the several other motor units that must be attached to these muscle fibers. Here is a histology slide showing you long cylindrical skeletal muscle cells with striations and nuclei, and here is the motor end plate where the axon terminals are getting close to the skeletal muscle fiber. So why are we talking about motor units? Why are we concerned with the nerve muscle functional units? It's because we want to talk eventually, especially when we move into chapter 10, about muscles as organs, as systems. And large weight-bearing muscles, which are called the prime movers for certain movements, especially the ones that move the thighs and the hips, they have really large motor units. And stimulation of a single motor unit may cause a weak contraction, but these large muscles would use multiple motor units to cause strong contractions and move more weight. Next, we're going to look at certain terminology, but we're looking at our muscles' basically response to varying stimuli. So muscle responses to varying stimuli depend on motor unit contraction, but also the frequency of the action potentials. So a muscle twitch is just a motor unit's response to a single action potential. But usually we have multiple action potentials, and if we change the frequency of stimulation as opposed to the strength of stimulation, we can create graded contraction. Now, action potentials do not add up on each other. They occur one at another or separately, but we can add up contraction. So if another action potential occurs right after the first one ends and that muscle hasn't completely relaxed, the frequency of the action potentials can generate more strength in that muscle by causing further tension summation. So pretty much what we're doing is causing changes in muscle contraction frequency by increasing the firing rate of motor neurons, thereby making sure that as soon as one action potential ends, another begins, and we get the summation of muscle tension. Because when the muscle receives identical stimuli of the same strength, and action potentials are always on the difference of about 100 millivolts, as long as those occur in rapid succession, the muscle doesn't have time to completely relax. So if we have more than just a twitch, for example, the second action potential, which causes more contraction, or a second twitch, you could say, would cause that wave summation. There's different types of wave summation. You have a single action potential, of course, which is a twitch showing no wave summation, all the way up to fused or complete tetanus. But unfused or incomplete tetanus is a little bit more common than fused complete tetanus. So what we're seeing is that the number of cross bridges formed between actin and myosin is responsible for determining the amount of tension produced by a muscle. So the length of a sarcomere is usually optimal when the zone of overlap between the thin and thick filaments is the greatest. And muscles that are stretched or compressed too greatly are not going to be able to produce maximal amounts of power. So we only want to use our motor units that are formed with a motor neuron and all the muscle fibers that are innervated by that same motor neuron if needed. So just to review, a single contraction is called a twitch. A muscle twitch generally has a latent period or a contraction phase and a relaxation phase, but a graded muscle response would not allow for that relaxation phase. It would allow for some variation in the muscle tension and cause summation, which would occur as a successive stimuli or as successive stimuli are added together to produce pretty much stronger muscle contractions as they build up. We don't really want tetanus. It would be the fusion of contractions to produce a continuous contraction. Complete tetanus, think someone lifting a car. That can be very damaging and cause a lot of fatigue in skeletal muscles, but it does occur. But we would increase the number of motor neurons involved, or increase the number of motor units, which use separate motor neurons, in order to increase the amount of activation in a muscle. And that increasing or adding motor unit process is called a recruitment. And this is actually even going to demonstrate or produce muscle tone, which is constant low-level contraction that allows for posture and stability. So we see some degree of wave summation regardless of what's happening, whether it's really active contraction or something that's just slight contraction that allows for you to sit up straight versus kind of falling out of your chair like Jello. So more rapidly delivered stimuli result in incomplete tetanus, and then the time between twitches would shorten in incomplete tetanus and would shorten or almost be non-existent in complete tetanus, because if you give stimuli quickly enough, you get complete tetanus, where the relaxation phase is eliminated and the sarcoplasmic reticulum doesn't ever reclaim the calcium ions during the contraction. Again, complete tetanus is something we want to try to achieve or shoot for because it's rare. It would be like someone picking a car up off of their child. So again, think superhuman strength. So much more common is kind of low-level wave summation or unfused incomplete tetanus, where we have a little bit of relaxation before we build up more tension because of a new action potential. So wave summation is important because it allows us to produce a continuous contraction, allows us to produce continuous active contraction of a muscle. It makes muscles useful. The force of contraction is going to be controlled by multiple motor unit summation. So if you use more motor units, you have more force. That recruitment, again, is just bringing in more motor units. And if you bring in more motor units or motor fibers into play, that would increase the strength of the contraction. All right, on the organ level, the contraction of muscle fibers, which are cells, and muscle organs, pretty much similar. So we're going to have a shortening of muscle cells, which will cause shortening of muscles as an organ. There's two types of muscle contraction as an organ. There is isometric and isotonic. Isometric contraction is when muscle tension builds, but the muscle neither shortens nor lengthens. And this would occur, for example, if the load is greater than the tension that the muscle is able to develop. And the example I gave earlier was pushing against a wall. So you can build tension while you're trying to push a wall over, but if you're not able actually to push the wall over, if your tension is not stronger than what you're trying to move, then no shortening actually occurs. Isotonic contraction is when muscle tension builds and the length of the muscle actually changes, that we get shortening. There's two types of isotonic contraction, but the key point to take away here is that during isometric contraction, the muscle doesn't shorten or lengthen. During isotonic contraction, the muscle shortens or lengthens. So we get movement, we get length change with isotonic that we don't see with isometric. So with those definitions, spend a minute figuring out which one of these is isometric and which is isotonic. In example A, we have the initial state of the muscle here on the left, and then the muscle over here shortens. In state 2, on the right, we have the initial state on the left, and the final state on the right, we see neither lengthening nor shortening. So one is isotonic, one is isometric. See if you can figure out which one. If you said that the one on the left was isotonic, you are correct, and the one on the right is isometric. All right, let's talk more about isotonic contractions where we see actual shortening or lengthening of the muscle. The two types are concentric and eccentric, which match with either the shortening or lengthening that is going to be associated with isotonic contractions. Concentric isotonic contractions are when the muscle shortens in order to do work, such as when you pick up a book or if you kick a ball, whereas an eccentric isotonic contraction is where when the muscle contracts, it lengthens. This is not as common, but your calf muscle or your gastrocnemius, when you walk up a hill, actually lengthens as it contracts. Next, let's talk about muscle tone, which was mentioned when we were talking about graded summation or responses. Muscle tone is just slight muscle contraction that stabilizes bones and joints. So even when you're relaxed, your skeletal muscles are always slightly contracted. This is important because it keeps your muscles ready to respond, firm and healthy, and it's due to spinal reflexes. So again, motor neurons that are activating one motor unit and then another. Responding to their activation, there are stretch receptors in muscles and tendons that kind of let your body know when muscle activation should take over from muscle tone and further motor unit recruitment is needed. So muscle tone itself does not produce active movements. It just produces kind of stabilization to create healthy, stable, and slightly contracted muscles. Our next topic is going to be muscle metabolism. So we're going to look at how do muscles generate energy for contraction. The energy for contraction requires ATP, so this will be an active process. So make sure that you go back and you're familiar with the anatomy and the physiology of muscles, review videos, and then finally come watch this last part of the Chapter 9 video lectures.